I'm Rick Lifton, the president of Rockefeller University, and it's my pleasure to welcome you all to tonight's program. This is the 14th annual presentation of the Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. Your presence is a fantastic show of support for an award conceived by two cherished members of the Rockefeller community. When Paul Greengard received the Nobel Prize in Medicine in 2000 for his crucial discoveries about the mechanisms of signal transduction in the brain, he and his wife, Ursula von Riedingsvard, donated the monetary share of his Nobel Prize to establish tonight's prize, which is named in memory of Paul's mother, who tragically died giving birth to him. Their vision and generosity inspired Friends of the University to provide additional funding, so in addition to the prestige of this award, this prize includes a $100,000 honorarium to the recipient. Paul and Ursula created this award based on their observation that women were not being recognized at a level commensurate with their scientific contributions. Paul was determined to use his prominence as a Nobel laureate to address this issue by drawing special attention to women scientists doing spectacular, groundbreaking research. The Meister Greengard Prize honors women who have made major contributions to biomedical research, with contributions who have made transformative discoveries to diverse fields, including cancer research, immunology, genetics, biochemistry, and neuroscience. Discovery by the prize recipients are among the most remarkable of the past 60 years by men or women. To celebrate the 10th anniversary of this prize, the wonderful science writer Evelyn Strauss, who is with us here tonight, profiled the careers of the first decade's awardees. These profiles were published as a book that highlights the career experiences of some of the world's greatest scientists. Copies of the book are available in the lobby, and I encourage everyone to pick one up after the program. Tonight, we are thrilled to honor JoAnne Stubbe with this year's prize. Dr. Stubbe was chosen by members of the prize selection committee, a group of esteemed scientists that includes 4 Nobel laureates. Please refer to your program for a list of this year's selection committee members. As we will go into later in detail in the program, Dr. Stubbe is recognized for her pioneering work illuminating the novel and unexpected chemistry by which the enzyme ribonucleotide reductase makes the building blocks for DNA synthesis. Each year, the Meister Greengard Prize is presented by a woman who, like the prize recipient, has had a remarkable career and whose contributions to her field have benefited society. It's my pleasure to now introduce tonight's presenter, Dr. Paula Johnson, the president of Wellesley College. Paula obtained her undergraduate degree from Harvard and Radcliffe, and then stayed on at Harvard for her MD and Master of Public Health degrees, and then completed her residency in internal medicine and fellowship in cardiology at Boston's Brigham and Women's Hospital. Paula became interested there in the striking differences in the manifestations of coronary artery disease and other diseases in men compared to women. And this ultimately resulted in her founding and serving as the executive director of the Connors Center for Women's Health and Gender Biology at Harvard. At the Connors Center, she led efforts to both undertake cutting-edge research and translate it into improved clinical care for women. She concurrently served as chief of the Division of Women's Health at Brigham and Women's Hospital and was a revered physician and teacher at Harvard Medical School as well as on the wards. Paula's vision, research, and ability to lead at the intersection of education, healthcare, and public policy have earned her key leadership roles in the local and national arenas, including terms serving as chair of the board of the Boston Public Health Commission and as a member of the National Institute of Health Advisory Committee on Research on Women's Health. She's a member of the National Academy of Medicine and the American Academy of Arts and Sciences. Not surprisingly, based on her extraordinary record of accomplishment, teaching, and leadership, in 2016, Wellesley College came knocking at her door, which resulted in her being named the 14th president of Wellesley in 2016. Paula has combined insights from medical science, psychology, and other fields to derive better models for investigating and treating women's health issues. And she has trained a new generation of physicians and scientists who are implementing these approaches. With her move to Wellesley, she is now overseeing the education of young women who will be tomorrow's leaders. On a personal note, I'm delighted to say that I've known Paula for more than 30 years as we served together as residents in internal medicine at the Brigham quite some time ago. I'll just tell you that you learn a lot about your fellow residents through the demanding and often trying times of residency. And Paula was memorable for not only being an extraordinarily talented and dedicated physician, but also for her exceptional grace under the often most trying circumstances of internship and residency. Add in her remarkable warmth and humanity, and it's easy to see why she is such a great physician as well as an educator and leader. I'm delighted to have Paula with us this evening. Please join me in welcoming Dr. Paula Johnson to the stage.
Thank you, Rick, for that really kind and lovely introduction and for your gracious welcome to the Rockefeller University. And it's so special to be here with you this evening. Your leadership was truly so important to me all those years ago. What Rick didn't say was that he was also my chief resident when I was a junior resident. And those are very rocky times. You do go through a lot, and your chief resident is somebody who you often turn to for that leadership, that three-dimensional leadership, and without a doubt, Rick provided that in the most wonderful and outstanding way. And what I really love to think about is to know that your leadership at the Rockefeller will help to advance science in extraordinary ways. So Rick, thank you. I want to thank members of the Meister Greengard Prize Selection Committee for inviting me to participate in the presentation of this award to Dr. JoAnne Stubbe, about whom I will have much more to say. I also want to give great thanks to Lulu Wang, a member of the Rockefeller University Board of Trustees and a beloved Wellesley alumna, and to all my Wellesley friends here today. So you've heard the origins of the Meister Greengard Prize, and Dr. Greengard and his wife, the sculptor Ursula von Riddingsvard, founded the Meister Greengard Prize in 2004 and funded it with Dr. Greengard's Nobel honorarium. They did so both to honor Dr. Greengard's mother, for whom the prize is named, and to raise the profile of outstanding women scientists who, as Dr. Greengard was very clear to say, still faced discrimination. Women had received at the time only 7 of the 184 Nobel Prizes awarded in physiology or medicine at the time that the prize was created. Since then, 5 more women have received this honor. Now, that being said, women still face tremendous obstacles across professions. Not a single Nobel Prize in any field has gone to a woman in the past 2 years. And the Nobel Foundation's board has expressed concern and has said that it will be taking measures. So for all that remains to be done, we have much to do, but we also have much to celebrate, and that's why I'm so thrilled to be here this evening. Tonight, I'm honored to present the Meister Greengard Prize to JoAnne Stubbe. Now in its 14th year, the Greengard Prize is widely recognized as the preeminent international award honoring outstanding women scientists. Tonight's celebration reminds us of both what we stand to gain, but also what we stand to lose. Women make up roughly half of the global population, and the world simply cannot afford to squander their many gifts. In her singular accomplishments, Dr. Stubbe is a testament to this basic truth. A professor at MIT since 1987, she was the first woman to receive tenure in the chemistry department and is currently the Novartis Professor of Chemistry and Biology Emeritus. Through her groundbreaking research She has transformed our understanding of what Paul Greengard has called complex chemistry underlying the biology of life. She did so by shedding light on the building blocks of DNA. This work has had profound impact on the science of cancer therapies. Notably, it's led to new applications for the anti-cancer drug gemcitabine, used to treat carcinomas that include pancreatic cancer, breast cancer, and non-small cell lung cancer. In collaboration with John Kosarich, she has also greatly advanced our understanding of bleomycin, an antibiotic commonly used to treat cancer. As I prepared for tonight, I was also intrigued to learn of another longtime collaborator, one who is less well known, but no less important. His name is Dr. Meister Stubbe, and he is, well, there is really no other way to put it, he is a dog. Dr. JoAnne Stubbe's dog. So please don't leap to conclusions. You should know that Dr. Meister Stubbe regularly attends lab meetings, and he is an avid follower of laser pointers as they move across research presentations. And I'm afraid that I can't tell you much more than that, but he does have an email address, and his picture is on the lab's website. So if you have more questions, you know where to reach him. But truly, collaborations aside, and on a more serious note, JoAnne Stubbe is a role model for women in a field that has far too few. She models the very best of what science has to offer. Not women's science, but science writ large. By whatever measure you choose, she is extraordinary. This alone is tremendously inspiring, but she also stands for something more. She reminds us of the power unleashed when women have a chance to flourish. JoAnne's talents are remarkable, as are her accomplishments, but the opportunities that came her way should not be. All women should have the opportunity to reach their full potential. All women deserve the opportunity to thrive. This conviction is what drives me, what's driven me throughout my life, and I trace this to the inspiration of two very different women, both of whom influenced me in profound and enduring ways. The first is my grandmother. Some of my most wonderful childhood memories are of spending time with her in our four-family home in Brooklyn, New York. She was warm and exuberant, a true force of nature. She worked in a factory, but that didn't stop her from saving her pennies and traveling to Europe. She loved her life, and she loved me. Then when I was 8 and she was about 60, something changed. She became listless and withdrawn. She stopped eating. Her care became all-consuming for my family. Now, many of you probably recognize the signs, but back then it took what seemed like forever to get a diagnosis. And by then, she was in a deep downward spiral. She had been hit by a deep depression, and sadly, she never recovered. The tragedy of this delayed diagnosis and the lack of science that was available to treat depression had a lasting impact on me. It sparked an aspiration to keep such things from happening, and it set me on a course that ultimately led me to my beloved college mentor, the late Ruth Hubbard. A bit of background for those not familiar with Ruth's work. As the first female biology professor to be awarded tenure at Harvard, in addition to her outstanding science, she became a powerful feminist voice in and for science. She made explicit something that others had known but not articulated, that science had made men the norm, both their bodies and their ideas. And much of what was deemed scientific truth was socially constructed and really deserved re-examination. One especially memorable experience was the independent study that I did with Ruth and the late Stephen Jay Gould my junior year. At the time, the field of sociobiology was ascendant, going virtually unquestioned in many quarters. Ruth and Stephen challenged this and pushed me to challenge it. This lesson went far beyond the academic debates of the time. It taught me that I had a duty to question accepted beliefs, that it was not only my right, but it was my responsibility. I first met Ruth Hubbard in the spring of my freshman year. I enrolled in her now legendary Bio 109, Biology and Women's Issues. It was such a deceptively simple name, I could not imagine how it would change my life. Ruth didn't set out to train disciples. Rather, she taught us to approach the world with the same spirit that she did, to explore the larger forces that shaped scientific work. She impressed on us that our ideas mattered, that we mattered. She did this in large part by showing us that we mattered to her. And if she, with all of her brilliance, took us seriously, who were we to question that? It's now been more than 40 years since I first stepped foot in Ruth Hubbard's Harvard classroom, and another 10 since I watched my grandmother's light fade. But regardless of the passage of time, these two women have been with me in spirit every step of the way. They spurred my research into the myriad ways that women are poorly served by science and medicine in our health system. They paved the way for the insight that women are far too often misdiagnosed across diseases, and that this often occurs because their symptoms are not what we call typical. Now, this is code for saying they don't look like men's, because now think about it. Why on earth would symptoms that are experienced by over 50% of the population be considered atypical? Ruth and my grandmother's legacies live on in the Mary Horrigan Connor Center for Women's Health and Gender Biology at Brigham and Women's and Harvard Medical School, which I was privileged to found in 2002. It's where science is advancing our understanding of the impact of sex, who we are biologically, and gender, who we are because of our social environment, how they impact health and disease. A little over a year ago, I embarked on a major transition. After 30 years in medicine and public health, I became the president of Wellesley College. Now, this move surprised some, but to those who know me best, it made perfect sense. It was a logical step in my lifelong quest to advance health and the health and well-being of women and to improve the lives of women. Far too often, we leave women's lives to chance. This insight was at the heart of my work on women's health, is now at the heart of the work that I pursue and aspire to do at Wellesley. How do we assure that all women have a chance to thrive? How do we make this the norm instead of the exception? I think of Paula Greengard's mother who died in childbirth, something that still happens far too often in the 21st century and has in fact been increasing in the United States. I think of my own mother who graduated from college at age 56 and in doing so fulfilled a lifelong dream. She did a remarkable job with the cards that she was dealt, but what if those cards could have been changed? What more might she have done? Even young women who seem to have everything can still too easily stumble if they don't get the right support. My own Harvard experience was outstanding, but I know of many, way too many women classmates who never found those crucial mentors or friends. You can plant a seed in fertile soil, but it also needs sun and water. At Wellesley, we seek to provide optimal conditions for young women to flourish in all of their diversity. And while medicine is not a typical route to a college presidency, it's clearer every day just how well it prepared me. I think especially of how my years in medicine and science taught me to lead with questions and to listen intently to their answers. The good physician approaches each patient with a sense of humility, deeply aware of the limits of her knowledge. We listen for the unexpected. We don't allow our working assumptions to blind us to new information. I learned to look beneath and beyond statistics to the person in front of me. Because as Rafia Zachariah recently wrote in the New York Times in her piece on women's empowerment, numbers never lie, but they do omit. At Wellesley, as we educate the next generation of women leaders, we also seek to ascertain the conditions in which women flourish. Like great scientists, we iterate. We experiment, evaluate, and try again. We do this over and over, and we make progress. So last year at this time, Girls Who Code founder and CEO Reshma Saujani stood before you as I am tonight. It was just days after the November's US presidential election. She talked about how hard the past week had been, how there hadn't been a day that she had not cried. I know just how she felt. On the night of the election, Wellesley hosted a huge gathering. It was about 3,000 people. We expected it would become a victory party for one of our most eminent alumni. We looked forward to one of our own breaking through what she had called the highest, hardest glass ceiling, and to celebrating the election of the United States' first woman president. Well, that's not what happened, and we're still struggling to navigate the strange new world that we have. But here's the thing, for all the pain of this enormous setback, we have also seen an explosion of activism and energy. The Women's March was the largest single-day protest in US history. More women are running for office. More women are speaking out, and increasingly their voices have impact. Just ask Harvey Weinstein. We stand on firm foundations, thanks to the work of countless women and men like Paul Greengard, who understand that when women take their rightful place, the whole world benefits. Tonight, we celebrate JoAnne Stubbe and her stunning accomplishments. At the same time, we are reminded of all that needs to be done to assure that the next JoAnne Stubbe has a chance to reach her potential. And I should say the next JoAnne Stubbes. And it goes far beyond historic moments where we break through glass ceilings, important though these are. It means attending to the structures that hold women back and strengthening those that move them forward. The stakes have never been higher, and the challenges are great. And yet, when I look around this room, I see a far brighter future. And JoAnne, you embody all that we can achieve. In that brighter future, you are our North Star. Thank you.
Thank you, Paula. What a moving tribute and beautiful talk. Thank you so much. I would now like to introduce JoAnne Stubbe, this year's Meister Greengard Award recipient. Dr. Stubbe is the Novartis Professor of Chemistry and Professor of Biology Emeritus at the Massachusetts Institute of Technology. So while DNA is the messenger of heredity in every cell on this planet, and stores all of the instructions to create and sustain the life of every organism, It is now widely believed that the first self-replicating nucleic acid on Earth was the related molecule RNA, not DNA. There are many lines of evidence for this. One particularly intriguing piece of evidence is that all forms of life synthesize the building blocks for RNA polymers from scratch, while all of the DNA building blocks, the deoxyribonucleotides, are made as a simple derivative of RNA, simply by removing a hydroxyl group from the ribose sugar of ribonucleotides. So while this superficially seems easy, just removing one oxygen and a hydrogen from an RNA precursor, the reaction mechanism was a puzzle for decades, in part because the mechanism turned out to be both unexpected and astoundingly complex, with many surprises on the way. This incredible chemistry was in fact discovered by tonight's honoree, JoAnne Stubbe. JoAnne's interest in science grew out of a love of nature and an unstoppable intellectual drive. She spent childhood summers at her family cabin on a lake in New Hampshire, fishing, swimming, and catching bait to make money, I'm told. She was known to be passionate and intense about diverse interests, which included bridge. As a 9-year-old, she was playing competitive bridge, the Boston Celtics, and tennis. And true to form, when JoAnne went off to college at the University of Pennsylvania, she carried diverse interests there as well, in art history, in math, and in chemistry. But she gravitated toward chemistry because of the appeal of its logic and the insights that could result from understanding the intricate details of chemical reactions at an atomic level. She earned her undergraduate degree and headed to the University of California at Berkeley for graduate school in chemistry in 1968. I dare say there were likely very few other women in the entering class in chemistry at Berkeley in 1968. At Berkeley, JoAnne became interested in the chemistry of biological systems. At that time, chemists viewed biology with suspicion and skepticism in general, and even disdain in cases, in part because the field was not yet generally yielding information at a molecular or atomic level. The chemistry department, for example, would not give her chemistry credit for a biochemistry course that she took from two really preeminent enzymologists, Dan Koshland and Jack Hirsch. Dan Koshland, in fact, as some in the audience would know, received the Lasker Award for Special Achievement, an award that now carries his name. So it's quite remarkable that they were unwilling to give JoAnne credit for taking a course from Dan Koshland. But Dr. Stubbe, nonetheless, was drawn to the nascent field of mechanistic biochemistry, fascinated by the idea that one could understand at atomic level detail how enzymes work and how small molecules can block these functions. After her PhD and postdoctoral fellowship, JoAnne was recruited as an assistant professor to Williams College, which came with a heavy teaching responsibility. The experience of covering subjects that were well outside her area of expertise made her realize just how much she wanted to work at a research institution. So remarkably, she took another unorthodox step. She left her faculty position at Williams to again become a postdoctoral fellow, this time at Brandeis University with Robert Abels, a world expert in mechanistic enzymology. This training shaped her future professional path, and it propelled her into a research position in the Department of Pharmacology at Yale School of Medicine. There, JoAnne made a breakthrough discovery on the mechanism of ribonucleotide reductase, the enzyme that converts RNA precursors into DNA precursors. Characteristic of JoAnne, many laboratories had tried to understand this mechanism, and many careers had in fact foundered on the shoals of what seemed an insolvable puzzle. Senior members of the field warned her that it was professional suicide to pursue this project. This did not slow her down. In fact, her work on ribonucleotide reductase continued for many years, carrying her from her position at Yale to University of Wisconsin, and then to MIT, where she has been since 1987. The answer, JoAnne discovered, involves molecules called free radicals. Free radicals are famous for their reactivity. Each one carries a single electron and desperately wants to pair up with another electron. As a consequence, they can wreak havoc on other molecules they encounter, including crucial ones inside cells like proteins, lipids, and DNA. But JoAnne showed that ribonucleotide reductase manages astoundingly to control free radicals and to harness their power to perform the catalytic step, replacing the hydroxyl group of RNA with a hydrogen found at this position in the DNA backbone. But in addition to this surprising reaction mechanism, the reaction is incredibly complex, involving the formation of a stable free radical on one protein that is then transferred to a series of cysteine residues in the catalytic domain, and then not to the carbon in the RNA that loses the hydroxyl group, but to the carbon next door. It's no surprise that this was not figured out prior to JoAnne. The radical drives the loss of the hydroxyl group of ribose and is eventually cycled back to its starting point, a truly astonishing reaction cycle. Dr. Stubbe's amazing research has had many important clinical applications. Ribonucleotide reductase is an important target in cancer, and insights from Dr. Stubbe's work has contributed to the development of gemcitabine and clofarabine, potent anti-cancer drugs used in a variety of pediatric and adult malignancies. Her work has also provided insights into the mechanism of another cancer drug, bleomycin, a natural product which cleaves DNA. In work unrelated to free radicals, she has probed how certain bacterial enzymes create large polymers that possess properties of plastics, yet are biodegradable. These polyesters might one day replace oil-based materials and thus ease the pollution burden generated by traditional plastics. JoAnne is an elected member of the National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. In addition to the Meister Greengard Prize, she has received many other accolades, including the National Academy of Sciences Award in Chemical Sciences, the Kaiser Award from the Protein Society, the Nakanishi Prize of the American Chemical Society, and in 2009, she was awarded the National Medal of Science from President Obama. We're delighted that a number of JoAnne's scientific colleagues are here to share this occasion with her. Suzanne Walker from the Microbiology and Immunology Department at Harvard Medical School, Elizabeth Nolan from the Chemistry Department at MIT, and Julia Page, a former MIT undergraduate who worked in JoAnne's lab and is now an MD PhD student at Harvard Medical School. Dr. Stubbe's astounding career and myriad discoveries are a source of inspiration for scientists of all genders around the world, and we are absolutely delighted to present her with this year's Meister Greengard Award. And now, JoAnne, would you please come to the stage, and Paula, would you please come up with us? Why don't you come over here, and we'll just read from this.
I got this.
Okay. Congratulations. This is what you're coming. JoAnne Stubbe, you're being honored for your discoveries in biochemistry, which have revealed unanticipated aspects of enzyme mechanisms and free radical chemistry, thus illuminating new avenues for improving strategies to fight cancer. By showing how the enzyme ribonucleotide reductase exploits free radicals to create the building blocks of DNA, you have unveiled nature's power to harness potentially destructive chemicals and direct them to perform complicated reactions with exquisite specificity. Your work has revealed how certain anti-cancer drugs interfere with this enzyme and disrupt other activities that are essential for life. You have set an exemplary standard for the scientific community by establishing innovative collaborations, devising new methods, and demonstrating that relentless curiosity can fuel an extraordinarily productive and satisfying career.
It is my honor this evening to present Dr. JoAnne Stubbe the Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the Distinguished Selection Committee of the Meister Greengard Prize and all of us here this evening, we congratulate you on this award.
Whoa, I should hire these people to introduce me all the time. You guys did a fantastic job. So I don't have that much to say, but let me just start by saying thanks. Thank you so very much for this amazing honor. Looking at the list of previous awardees and reading or seeing the videos about their lives and their contributions, I really am very humbled to be included in this group. And every morning I wake up and I pinch myself to see if it was really real. Anyhow, I also like to thank first and foremost Paula Greengard and Ursula von Reitingsard. Your commitment to spotlighting outstanding women scientists is inspirational and extremely important. And I think it's even more important now in the current political climate. I would also like to thank the selection committee for this award, who chose me for this award, because from my experience, I know that it is fun to be involved in being undergoing a selection of this type, as you get to discuss and learn about amazing accomplishments of many of the nominees. So I hope you had a good time. I am also grateful to Paula Johnson, who, for introducing me. That was a really inspirational speech. Thank you so much in presenting me with this award. And I have written down that, unbeknownst to her, but I told her before right before I'm giving this speech, that this is a special gift to me because I have had a very long time indirect connection and fondness for Wellesley through my friendship with Diana Chapman Walsh, who was president of Wellesley for many, many years. And she's married to one of my best friends, Christopher Walsh, who is also an enzymologist, so thank you. I also want to thank Evelyn Strauss for all the work you did. That was really, really good. Anyhow, thank you so much for making me also into a real person. Often, I've been called the scientist, okay, which I think is probably an insult. But anyhow, it's great that you make me seem more human. And in the last few minutes, I just want to talk very briefly about three things, some of which have already been touched on. But as you've already heard, that Paul Greengard gave money from the Nobel Prize to honor his mom. That is really special and very touching. I think it brings tears to your eyes. I know how lucky I've been to have had a mother and a grandmother as role models throughout my childhood. My mother and Grammy, in retrospect, had an amazing effect on my resilience and ability to cope. They also instilled in me a passion to learn, to do my best, and to be satisfied with whatever the outcome actually was. I also want to say that while today we're women-centric, my dad also played a very important role in my strength and resilience. He was a mathematician, and when he learned I was going to graduate school in organic chemistry, he had me buy him an organic chemistry book so he could learn how to talk to me. The second thing I'd like to briefly say is that, because I thought it was pertinent to people in this audience, is I was on the first committee tasked to report on the status of women at MIT in the School of Science that took place in 1995 and 1997, to 1997. Many of you probably know about this as the Hopkins report. She was one of the women and clearly the leader of getting this all to work. Our committee actually had access to data The university gave us hard data, and at MIT, data matters. So with this data and the leadership of Hopkins and the support of the dean of the School of Science, who was then Bob Bergeneau, and the president of the university, who was Chuck Vest, a report was written and identified and then tackled, MIT tackled issues of inequities in salary, space, and resources, among other things. Changes were made subsequent to this in a second report that was carried out in the School of Engineering, and procedures of hiring changed, and MIT has really become a much more family-compatible institution with daycare and family leaves, et cetera. I would say MIT has been in the forefront in getting universities to think about the problems. I think the big issue of marginalization of women faculty subsequent to tenure, in my opinion, has still not been resolved. For me, the experience of serving on this committee and meeting and befriending many of the 17 total women faculty members in the School of Science out of 280 men made me realize, actually, that I was not alone. So it was an interesting experience. Third is, as you already heard from Dr. Lifton, when I was asked by Amanda if I had a few guests I wanted to accompany me to celebrate this award, it took me a few minutes, only a few minutes, to decide to invite three generations of young women, three young women from three different generations that have really enriched my life tremendously. I am extremely proud of all of you. Julia, as you heard, is an MD PhD student. She had me in 2 classes in biochemistry. I used to use her answers to grade the exams. She's now an MD PhD student at Harvard Medical School. Liz Nolan, she's the next generation, is a recently tenured colleague at MIT with whom I teach. I love teaching with Liz. I talk a lot of science, as we have a lot of overlapping interests, and we also get ice cream at Toscanini's. For the third generation, I was delighted when Suzanne Walker, a faculty member, as you've already heard, at Harvard Medical School in microbiology and immunology, and an affiliate member of the chemistry and chemical biology department, was enthusiastic to participate. She has been inspiring to me, and as with me, has had an unconventional career. I love your science, Suzanne. I love talking to you about your science and science in general. And I thank you and your family for being bringing so much support for me. So I thank you all for joining me today in this celebration. It has meant so much to me, as does being the recipient of this very special award. So thank you very much.
That's just great. Thank you. So now why don't we step over here? We have the microphone. So thank you, JoAnne, for those wonderful comments. And now we're going to have some time for questions and answers. I think I'll lead off as we get mic'd up here. So,
hello. Can you all hear me? I always do that in class. Raise your hand.
So JoAnne, much of science is about taste, picking the right problem at the right time. How did you come to pick ribonucleotide reductase as your target?
Yeah, it's complicated. So when you described that I left Williams College and took a leave of absence and worked in Abel's lab. And the reason I went to work in Abel's lab is because he was interested in designing mechanism-based inhibitors. So I work on enzymes, so as a catalyst, everything in your body is catalyzed by an enzyme. You couldn't do anything without hundreds of enzymes. And if you understood how you converted some substrate into some product, you understood it from a chemical perspective, could you design something that would specifically inhibit the enzyme if that enzyme was going to be a target of interest? For example, it was unique to bacteria and not to humans. So I went to Abel's lab to think about mechanism-based inhibitors, and when I was there, a paper came out by Reichard and Scheuberg and Ehrenberg and Grassland that showed that the enzyme ribonucleotide reductase had a tyrosyl radical. So that's one of these highly reactive species that you can't control. I mean, in solution, it has a lifetime of a microsecond. And I read about this, and that was unprecedented, that there was a tyrosyl radical that would play a role in any reaction, never mind making the building blocks required for DNA. And at the same time, Abel's, whom I was working with, was a discoverer of how vitamin B12, coenzyme B12 worked. That also involves radical chemistry. And there was another ribonucleotide reductase that used B12. So immediately, as a chemist, I started thinking about radicals. You know, radicals are considered to be bad, uncontrollable. But why, given radicals with this new discovery of a tyrosyl radical and B12 being radicals, couldn't nature figure out how to harness the reactivity of radicals to do really tough chemistry with exquisite specificity? And so, and at the same time, there was a paper published right before I went to Abel's lab where people had reported inhibitors of ribonucleotide reductase. It's a major target to design anti-tumor agents. Why? Because cancer cells divide faster than normal cells. This isn't all completely true, but if you can inhibit the cancer cells uniquely and not the normal cells, you prevent your DNA from replicating, and you have a potential anti-tumor agent. Anyhow, somebody had reported molecules that could inhibit ribonucleotide reductase, and they had no idea how they worked, and they proposed something chemically that was silly. And so those two things together, again, that's why being trained as a chemist was great. I was at the right place at the right time. And so I put those two things together, and that's how I began. The chemistry was fascinating, and it was important in terms of human health.
That's great, thank you. So this free radical chemistry in the enzyme reaction center seems amazing, since they are typically so incredibly short-lived, and yet the tyrosyl radical can hang out for days, and then it gets transferred in this Rube Goldberg-like mechanism to successive cysteines and then the adjacent carbon. How does it manage to be protected from reacting with everything in its path?
Well, I mean, so if you generated a normal radical, a tyrosyl radical in the middle of a protein, it would get reduced, pick up an electron and a proton really fast. So somehow, and it travels, in most chemistry, it happens over 2 or 3 angstroms, 4 angstroms. This reaction happens over 35 angstroms. So to do that, you have to have intermediates along the pathway, and that's where you have these hopping radicals. So the question is, how do you avoid killing yourself? These radicals are dying to pick up an electron and a proton. They're dying to be reduced. They're as hot as the hottest oxidant that we have in biology in general. These things are that hot. So how does nature control that? The answer is we really don't know completely. But you know, in the place where you have the stable radical, it's somehow protected by the amino acid side chains to prevent it from getting reduced and picking up a proton. And then we've shown in the last few years that this long journey you have to take is uphill. Okay, so that means you're going uphill, stepping uphill, uphill. You get less and less and less and less and less of it. So you don't make very much of these intermediates along the way. Okay, and so, but you need to make enough so that when you get up here, you can make your building blocks. And then you got to go back, all the way back downhill to get to the starting material. So our model is that nature has designed this to be uphill because there's not enough of these intermediates. They're not long-lived enough to be able to be trapped by normal reactions you would find at proteins. And so that's what we believe her design is. But why would nature, maybe somebody here, you're all biologists, maybe somebody here can tell me why would nature ever design something with so many reactive intermediates over 35 angstrom distance? I mean, nobody believed it was true. That's why I started working on it. Okay? But I think the data, we have pretty good evidence for it now. So I don't know. I don't really know the answer. But you can protect the stable tyrosyl radical. It's very hydrophobic. There's nothing that can donate, no amino acid side chains that can donate an electron.
It's an amazing story.
Yeah.
So it seems as if one of your consistent hallmarks has been marching to your own drummer. So going to chemistry graduate school at Berkeley in the 1960s, leaving a faculty position to go back to being a postdoc, picking a project that people warned you was likely career suicide. So, and yet here you are today with all of us, for which we're very grateful. But my question is, what advice would you give to young scientists in the audience today?
I don't know. I've always followed my passion, and you live with it. You do the best you can, and you live with the results. Like, I love tennis, right? But I'm not playing in Forest Hills. Okay. So I mean, I used to get clobbered, but I mean, I would go out and practice every day because I really, really liked it. So, I mean, you just have to live within what your abilities are. And I think I was extremely lucky, chance of being in the right place in the right time and having things come together all made a difference. I work really, really hard. I don't really work. I love what I do. So, I mean, I don't know. If you're lucky enough to be able to find and pursue your passion, then you'll have a good life. I've had a great life.
That's great. Thank you. We have time for a few questions from the audience. There are roving microphones, so please, if you have a question, raise your hand and a microphone will come your way. Lulu?
Thank you, Dr. Stubbe, for a very inspiring presentation. I have a question to follow up on what Rick was asking about young people. With funding now so difficult for research, how do you encourage young scientists to stay the course and try to find that elusive research dollar? And if they don't, how else do you encourage them?
So I'm like half deaf. Can you repeat?
So how do you encourage young scientists with the challenges in funding, and if that becomes a tough road for them, how do you encourage them to stay on?
Everything is life is tough. I don't know. I mean, you can't sit around. No, I mean, really, you can't sit around and worry about funding. I mean, now the funding's bad, but it goes like this. You got to figure out how to squirrel your money away on the side to pay for the times when you lose your funding, so you can keep supporting your research. You got to. I don't know. I mean, I think if you are unsuccessful at what your passion is, then you've got to find something else to work on. What can I say? But if you pursue your passion and you're good at it, you might not do it at this level, but you could do it at this level or this level or this level. As I'm retiring now, I'm thinking about going back and working in somebody else's lab because I love working at a bench. So that's one. Yeah, I can't. I'm too old now.
Other questions?
So that probably didn't answer your question, but.
Any other questions in the audience? Yes, over here, Leslie, thank you.
Sorry, this is going to be provocative. So the men who told you that women can't come to their lab to train, they don't take female scientists, did any of them live to see your great success, the national medal? And what would you say to them?
No, they're all dead.
That's all we needed to hear.
I mean, let's put it positive. I mean, times have changed, right? So if you look at what what this women's report did at MIT, it really made a difference, I think. We still have a long way to go, but it made a huge difference. And I see, we have these young people sitting in the front row who are all outstanding scientists. So times have changed since those days. And I had a good time even in those days, right? I mean, the Grateful Dead used to play in, I went to the San Francisco Symphony. I had fun working in the lab. They used to dump tear gas on the campus, anyhow. So even those days were interesting.
We have a question here.
Given your vivacity and your enthusiasm, I'm curious, how many hours a night do you sleep?
Well, when I was young, not many. Then I went through a phase where maybe 5 years ago, where I needed a little more, but now I'm back in the phase where I get up and I still have like ideas in the middle of the night. I can't, it's like it doesn't, I walk around and I walk around. I want to do one more thing before I really retire. Cryo-EM. Cryo-EM.
It's come to the right place. Well, with that, I think we're at the hour. There's one more question over here.
Hi. How long was it before when you first made your discovery that you realized how important it was, and how did the importance evolve over time? My sense is you made this discovery.
Can you say it again?
As you made this discovery, what was the trajectory of realizing how important it was, and how did the importance evolve over your career?
I remember when I made this discovery. I remember it, and it was like the most thrilling moment. I can just feel the adrenaline surge up. So I was in the chemistry department. I was in the pharmacology department at Yale, and I took an NMR spectra, and there it was, a singlet. And it was inorganic pyrophosphate that didn't seem to have anything to do with the reaction, but all of a sudden, everything I was thinking about fell into place. And I knew it was really important because nobody could figure out how this worked. And it's withstood the test of time. So, there's nothing like making the discovery yourself. That's what we do. We do this because we like to solve puzzles and put all the pieces together, but we also do it to discover.
Well, I think we all appreciate why she has won this award, and her passion for science is an inspiration for all of us.